Hi, Brad. Thanks for joining me today. I really appreciate it.
Hi, thanks for having me. Happy to be here.
Absolutely. So, just to start off, what originally drew you to working on neglected or opportunistic infectious diseases rather than traditional pharma targets? And could you help define what makes a disease neglected and why that matters for drug discovery?
Sure. I was first exposed to both opportunistic pathogens and NTDs as a grad student and in my first postdoc. Most of that work was in African sleeping sickness. Neglected diseases or neglected tropical diseases. Coincidentally, Neglected Disease Day is January 30th. It's tomorrow.
Oh, wow.
Neglected diseases are diseases that disproportionately are endemic to low and middle income countries. And so the resources for infrastructure and clinics is low, but also the commercial interest for pharmacotherapeutics is low. I have a passion for working for the most vulnerable populations, and that's certainly one subset of vulnerable populations. Neglected opportunistic pathogens, on the other hand, are diseases that disproportionately affect individuals experiencing immunodeficiency. So that's not just AIDS, that's also cancer and solid organ transplant. So they're a little less endemic to the tropics, but also a vulnerable population. And because the number of people getting those diseases is small, there's also reduced commercial interest in discovering new drugs. In particular, the diseases I'm most interested in are not caused by bacteria, they're caused by fungus and parasites, which is not to say that antibacterial drug discovery is easier, but they have different hurdles. One of our hurdles in eukaryotic pathogens is that a lot of our machinery is shared, and so we have a lot of the same proteins. And so the challenge is if you find a drug target, you want a molecule that gums up that drug target in the pathogen and not in the patient, and that could be extra difficult, but an exciting challenge, I think.
Yeah, that was certainly something I was curious about in terms of why they're particularly challenging to treat. How does drug resistance shape the questions that your lab asks early on?
Well, early on, probably not too much. I'm trying to find new drug targets. There are groups out there that try to make a molecule better or defeat resistance with the same target. That's like another generation of the same type of drug. My lab, we try to find new targets. So up front, I'm not really worried. We're trying to help with drug resistance. Ideally, we would find a molecule that inhibits something else in the pathogen and maybe could be used in combination therapy and help down the line with resistance. But it's not at the forefront when we're thinking of new molecules.
And your lab works in both target based and phenotypic drug discovery. How do you decide which approach to use for a given project?
Oh, that's an interesting question. I have more of a background in enzymology, I guess, but I have some background in microbiology. So, target based drug discovery is deciding on a target from the pathogen, in this case, that you want to inhibit, and then cloning that and doing biochemical assays as opposed to phenotypic is growing cells. In my lab, I'm at an osteopathic medical school, and my students are wonderful, but they're medical students and they don't have a whole lot of time for research. So I do have some students who have some background in biochemistry and protein science, and then I have some students who maybe don't. And microbiology, I don't want microbiologists to come at me, but it's a little more forgiving. If you're off by when you're pipetting enzyme catalyst, if you're off by a few nanoliters, it can throw off the whole experiment. The fun thing about doing the biochemistry is that I can take the same target from different parasites or different pathogens and test them without having to have a menagerie of microbes in the lab. So there are some pathogens that are a little too well, some that are too dangerous to grow in the lab, but some that are maybe take a little more finesse than I'm used to. And so we don't grow the super difficult to grow ones in my lab. But we try to do both. So I do have some proteins and some microbes where we're taking to approach.
You've emphasized binding kinetics as an early screening strategy. What problem is this approach trying to solve compared to traditional potency metrics?
So, like I said, most of my targets are eukaryotic pathogens, and we have a lot of the same proteins. To find a reasonable molecule to advance to the clinic, you need selectivity for the pathogen over the patient, which is difficult to do. We might not look like it, but we share a lot of the same genetics as, say, a fungus. One of the ways to try to achieve that selectivity is through binding kinetics. Binding kinetics refers to how long a molecule binds to its protein target. Some molecules bind in a rapid equilibrium. Some bind for a while. And some of the decongestants that are long-acting have a higher residence time. Since I'm trying to kill pathogen cells, I'm looking for binding kinetics that are pseudo-irreversible, that bind and don't bind on a physiological timescale. If you're looking for irreversible inhibitors, the covalent irreversible, it's easy to find a molecule that covalently irreversibly binds to a protein, but it's less easy to find one that binds irreversibly to the one you want and not willy-nilly along the way. So we try to find molecules that bind preferentially in a pseudo-irreversible fashion to the pathogen targets.
I know that your projects don't rely on small molecule chromatography by any means, but this is generally a chromatography focused podcast. So I was curious, where can chromatography still play a role in your workflows?
So I did a lot of chromatography in the past. Right now, I rely on colleagues and commercial supplies of molecules to test, and my colleagues making me molecules. But certainly, those people are using chromatography for synthetic steps and purifying the molecules that we ultimately test in either proteins or microbes. My lab started a project recently, just getting into a little chromatography in my lab, where we're looking at pharmacognosy, which is trying to find drugs in nature. And since we're in Las Vegas, we've tried to isolate some compounds out of the desert flora here in the Mojave Desert, and see if we can find some molecules that haven't been tested. There are a lot more groups who have done a lot more than we have. We're really only starting that. But yeah, the pharmacognosy is important. There's a lot of anti-infective drugs that are natural products or natural product based. There are a lot of drugs that aren't anti-infectives that were first discovered in fungus or plants. So who knows what else is out there in plants we didn't even look at yet.
This might be a tricky question, but I'm curious how your past experience, including your chromatography work, did that impact how you think about biological systems in general?
Well, I've done some past work where not only looking at molecules from natural products and needing chromatography to separate those. With some groups, not my group, if you're trying to deconvolute a target, let's say I get a phenotypic hit and I have a molecule that is killing my parasite cells in culture. The downside to phenotypic drug discovery is that I don't know what its target is. Whereas the downside to target-based drug discovery is there's lots of other proteins and things that can intercept it in the pathogen and the body. If you're interested in deconvoluting a target, if I have a molecule that is inhibiting parasite cultures, one way to try to figure out what it's hitting is to look at the metabolomics of the parasite cultures and see what molecules are building up and what molecules are depleted. And that requires quite a bit of HPLC and mass spec.
Neglected diseases don't usually have a clear commercial pathway. How do you think about impact and success for these projects?
I have a passion for trying to help vulnerable populations. Certainly, commercial success is good, but my main priority is contributing to. It'd be great if we found a potential drug candidate. We don't have one yet, but even if we don't, the things that we learn and share with the community might lead to a discovery that can advance to the clinic. And so I consider that success.
And what role do the students play in advancing this kind of early stage discovery, especially when the projects are of an exploratory nature?
So like I mentioned, I'm in a medical school, and my students are medical students for the most part. And working on neglected diseases actually allows us to make a meaningful impact. Whereas if we were working on drug targets that all the major pharma companies were working on, anything we're trying to discover would be discovered before us. So that's nice for the students. A lot of medical students aren't getting a really deep understanding of the drug discovery process. I mean, they learn pharmacology, but they don't learn the drug discovery process. And so my students have a much deeper understanding of what that takes to get a molecule from the bench to the clinic.
Based on your experience, what skills should chemistry or biochemistry students prioritize if they're interested in infectious disease drug discovery?
There's so much to do in infectious disease drug discovery. I would say there's a lot of room for different expertise. So if someone is interested, they could work in microbiology or toxicology. If they're interested in protein science, they could study protein science and work in a biochemistry lab. If they're interested in synthesis, synthesis is a huge role in drug discovery, or separation science. There's room for everybody. I think it's important to be passionate about what you're doing and be interested in what you're doing. Maybe not 100% passionate, but actually be interested in what you're doing and be good at it, right? There's a lot of room for people wearing different hats in the drug discovery process. And I would prioritize doing what you like rather than what you think somebody wants you to do.
I think that's a great answer. And as a manager of people, I certainly see how that translates beyond science and just in the workplace in general. What scientific or technological advances are you most excited about for accelerating drug discovery against neglected pathogens?
Well, not to answer the cop-out answer, but artificial intelligence. There are many groups who are currently using AI where they feed biochemical data and cellular data of molecules that have inhibited or have helped treat cancer cells or pathogen cells. And AI can make predictions of what other molecules might work or what other molecules you have in the freezer that might work. So that's exciting. I mean, that should really accelerate drug discovery in many areas. I'm not sure how fast it will accelerate neglected diseases and opportunistic pathogens because those programs require large data sets. There's large data sets for cancer targets, but less for some of these parasites. But that's exciting. There's AI that helps with figuring out how to make molecules too, right? So if the first AI pass says, you need this molecule, and how am I going to make that? The second AI can say, why don't you make it this way? So the AI should really, it's going to be exciting to see in the years moving forward in drug discovery.
In terms of the AI application, are there any large data collaborations where people are feeding in their data sets to a larger database so that that database is able to use AI to draw some conclusions from that shared data pool?
You want me to plug somebody else's widget?
If you want to.
Collaborative Drug Discovery, or CDD, is such a platform. Their model is they have electronic notebooks and places to consume data and analyze data, but they do offer free data sets. So if you're working on malaria or sleeping sickness or some of these diseases, some of those data sets are open if researcher has deposited them in the open repository.
Yeah, I was just thinking back to some of what I've learned about protein folding and how DeepMind, Google had worked to crack that code and how it was such a big deal. But that was a big part of the application of AI in general. And I previously interviewed Dwight Stoll, one of the leading chromatography experts. He's written a column in LCGC for a number of years on troubleshooting topics. But yeah, it's all for him. A lot of the conversation focused on the pooling of retention data points and all of the cool current developments, but also the promise of what can happen when all of that data is aggregated and analyzed for patterns that people have missed.
Yeah.
But yeah, Brad, what else about any of the topics today would you like people to know about who are just learning about this pathogen work or neglected disease and drug discovery for the first time?
I feel obligated, like I said, tomorrow's NTD Day, raising awareness for some of these diseases. One of the diseases we work on is Chagas disease, which is endemic to South and Central America. But the pathogen is in southern United States. So some of the diseases are potentially closer to home than we think. And then certainly with the opportunistic pathogens, as a reminder, opportunistic pathogens disproportionately affect people experiencing immunodeficiency, which is not necessarily AIDS. It could be somebody on immunosuppressive therapy. One of the diseases we work on is cryptosporidiosis, which is a gastrointestinal pathogen that for immunocompetent individuals, it's unpleasant. There might be watery diarrhea for up to a week, but it's usually self-limiting. For people experiencing immunodeficiency, the antiparasitic drugs we have are not very efficacious because those drugs rely on the immune system to do half the work. Crypto is actually the reason if you go to a public pool, it says if you've had diarrhea in 14 days, don't come in because it's actually chlorine resistant. Some of these bugs are pretty interesting, I think, from just a science view, from an evolutionary point of view. Did that answer your question?
Yeah, no, absolutely. Yeah, I'm always curious. I do the best I can coming up with some questions ahead of time, but I always want to make sure that we circle back to anything major that you want to discuss and you want the audience to know. Will I be seeing you at any ACS events later this year?
Not this year. My group went to Pacifichem. Well,
Okay.
I met you at the Worm. I had some students at Worm, and then I had some students present at Pacifichem on trying to find new drugs for acanthamoeba keratitis and invasive candidiasis. So that was probably our last ACS meeting for a while. I don't foresee going this year, but hopefully next year.
Well, Pacific Chem is certainly a large budget investment, let's say,
Yeah.
to get there. And I mean, it's not every year, so it's a very cool opportunity, especially if you have something to present. If topics you're interested are being talked about, it's definitely an opportunity that I'm glad you got to take advantage of.
Yeah, it was my first time at Pacifichem. So yeah, I did enjoy it. It was a good meeting. The international meetings are always interesting to see, not just domestic work that's being done on different things, but across the globe.
Totally. And yeah, one of the reasons the larger ACS meetings in the fall and the spring are useful is because with the different cities and the different timelines people have for presenting work, large, it's always a mix of new people, but I certainly see a few of the same people at every meeting. But obviously, at an international event, that's going to be a lot of novel things and people you haven't met before and a lot of cool networking opportunities for sure. But Brad, I really appreciate the time today. Thanks so much. And certainly, if there's ever a project you're interested in discussing and coming on to share with the audience, I'd be happy to talk again.
Thank you for having me. It was great talking.